Hello and welcome to Meditut, the medical tutorials. Our motto is understand, learn, and give your best performance. As discussed in lecture 5 of Meditut, the medical tutorials, any chemical species which dissociates into ions is called an electrolyte. Sodium chloride being the typical example of an electrolyte, and there are many other molecules which qualify as electrolytes. Acids and bases, they're also electrolytes because they dissociate into ions. But in case of acids, the cation is always a hydrogen ion, and in case of bases, the anion is always a hydroxyl ion. But they're electrolytes because they dissociate into ions. This is a hydrogen atom. In the hydrogen atom, there is no neutron, there is only a proton in the nucleus. When the electron is lost, what is left is a proton. So hydrogen ion is basically a proton. While the hydrogen atom, I am saying the entire hydrogen atom, that is an equivalent of electron. It is not an actual electron, but it is an equivalent of electron. When the hydrogen is transferred, means an electron is transferred. For example, when NAD is converted to NADH, means it has gained an electron. When FAD is converted to FADH2, means it has gained 2 electrons. So H, the entire hydrogen atom, is an equivalent of electron, while the hydrogen ion is an actual proton. Hydrogen ion is an actual proton. So the acids, because the cation is always a hydrogen ion, so acids are proton donors. The bases have hydroxyl ion as the cation as an anion. So hydroxyl ion combines with the hydrogen ion to form the water. Means bases are proton acceptors. This is a general definition of acid and base. Acid is any chemical species that donates proton. Base is any chemical species that accepts proton. So acids and bases are basically electrolytes, and anything that donates proton is an acid, anything that accepts proton is a base. Strong acids and strong bases are almost completely dissociated. For example, if there are 100 molecules of HCl, it will be almost completely dissociated into H ions and chloride ions, maybe 99%, 99.9%. Means basically it is present in the dissociated form. Very small amount is present in the undissociated form. Similarly, the strong bases like sodium hydroxide, that is basically present as a sodium ion and hydroxyl ion. Very small amount is present as a undissociated base. So practically this reaction is irreversible. On the other hand, the weak acids and weak bases. Weak acids and weak bases are partially dissociated. For example, this is a weak acid. It dissociates into H ion and its corresponding anion. It is not completely dissociated. Suppose there are 100 molecules of this weak acid, maybe there are 50 H ions, 50 anions, and 50 undissociated acid. Maybe there are 30 H ions, 30 anions, 70 undissociated acid. So it is not completely dissociated, partly present as the undissociated form and partly present as the dissociated form. So this reaction is reversible. Suppose there is a deficiency of H ions in the surrounding medium. So when there is a deficiency of H ions in the surrounding medium, the forward reaction will be favored. Means the weak acid will be more dissociated if there is a deficiency of H ions in the surrounding medium. But if there is an excess of H ions in the surrounding medium, the reverse reaction will be favored. Means the weak acid will be less dissociated. What I mean to say is that dissociation of weak acids depends on the H ion concentration of the medium. And the H ion concentration, the hydrogen ion concentration of the medium is expressed as pH. pH is negative log of H ion concentration. Remember, it is the negative log of free H ion concentration. Suppose there are 100 molecules of this weak acid and it is 50% dissociated. So these 50 H ions will contribute to the pH, not the entire 100 acid molecules. Remember, this is a very important concept. The pH is negative log of free. I'm saying free, the free H ion concentration of the medium. So the dissociation of weak acid depends on the H ion concentration of the medium, or you can say the pH of the medium. It is inversely proportional to the H ion concentration and directly proportional to the pH of the medium. Why directly proportional? Because pH is negative log. More H ion concentration, less will be the pH. Very important concept. Inversely proportional to the H ion concentration, directly proportional to the pH of the medium. So the dissociation of weak acid depends on the H ion concentration of the medium or the pH of the medium. But at how much pH, how much will be the dissociation? So this mathematical relationship between the dissociation of the weak acid and the pH of the medium or H ion concentration of the medium is expressed as Handersen Hasselbalch equation. Handersen Hasselbalch equation is pH is equal to pK plus log of the concentration of anion upon the concentration of undissociated acid. Means how much is dissociated, how much is undissociated, that depends on the pH of the medium through this equation. In this equation, pH we have already discussed, it is a negative log of free H ion concentration of the medium. There is another term called pK. Now A here stands for acid because we are discussing the dissociation of weak acids. So A, small a. So pKa. pKa is the negative log of dissociation constant of the acid. See, every acid or every base has got some dissociation constant, which is a fixed quantity. So if we take the negative log of dissociation constant, that is called the pK. Negative log of free Hn concentration is called the pH. A minus is the anion and Ha is the undissociated acid. Now see here, because this is a reversible reaction, so this anion can combine with the Hn to form the acid again. Technically speaking, this anion is a base because the definition of acid and base, anything that donates proton is an acid, anything that accepts proton is a base. So technically speaking, this anion is a base. So this is called the conjugate base of this acid. For example, lactic acid is acid. Lactate is the conjugate base. Acetic acid is acid. Acetate is the conjugate base. Citric acid is acid. Citrate is the conjugate base, and so on. So A minus is the conjugate base of this acid. So we can say Handersen Hasselbalch equation is pH is equal to pK plus log of the concentration of conjugate base upon the concentration of acid. Conjugate base upon acid. Another important point I would like to clarify here is, I told you pK is a negative log of dissociation constant, pH is a negative log of Hn concentration. These are two different entities, these are two different terms, their definition is different, not same, they are two different terms. However, their mathematical value is equal when the weak acid is half dissociated. When the because the dissociation of weak acid depends on the pH. So as the pH changes, the dissociation also changes. So when the dissociation is half, it is 50% dissociated, the pH is equal to its pK value. Only their numerical value is equal. Otherwise, they are two different terms. Okay, so pK is negative log of dissociation constant and its mathematical value is equal to the pH at which a weak acid is half dissociated. Always remember it. Suppose this is a weak acid and it is present in a medium along with its salt. The weak acid gets dissociated. The salt also is dissociated because they have a common anion. For example, citric acid plus sodium citrate. So citrate ion is common. So because the anion is common, the dissociation of weak acid tries to decrease the dissociation of the base. And the dissociation of the base tries to decrease the dissociation of the acid. And they reach an equilibrium. So when they reach an equilibrium, there is some HIN concentration of this medium. Or you can say there is a fixed pH of this medium. So HIN concentration or pH of this medium is fixed. Right? Let us suppose we add an HIN from outside. Means we add an acid to this medium. There will be excess of HINs here. This reaction will be favored in the backward direction. But to keep the concentration of anions here, the salt will dissociate more. Means whatever extra HINs have been added, they are converted to the undissociated acid. Means the original HIN concentration remains constant. The original pH remains constant. Means the pH does not change even when we add an acid from outside to this medium. Let us discuss another scenario. Suppose we add an alkali from outside. When we add an alkali from outside, that will take up the HINs. And as soon as it takes up the HIN, the forward reaction will be favored. And to keep the concentration of anions constant here, the salt will dissociate less. Means whatever HINs have been taken up by the alkali, they will come by more dissociation of the weak acid. So again, the original HIN concentration or the original pH of this solution remains constant. So this is called buffer. Buffer is a solution of a weak acid and its base and its salt. It resists the change in pH. Let us suppose this buffer has a pK value of 4.76. For example, sodium acetate plus acetic acid buffer, or simply we can say the acetate buffer, it has a pK value of 4.76. So initially the pH of the medium is 4.76, which is equal to its pK value. Now we add an alkali from outside. As we add the alkali from outside, the pH should not change. It should not increase because it is a buffer. But actually speaking, the pH changes. The pH changes, but the change is very, very small. Even when we add a large amount of alkali, the change in the pH is very small. So we keep on adding lots and lots of alkali. The pH keeps on getting increased till it reaches 5.76, and after that it will suddenly increase. Similarly, when we add acid to it, the pH decreases, but the decrease is very, very minimal. It decreases very, very slowly till it reaches 3.76, and after that it will suddenly decrease. Means between the pH of 3.76 and 5.76, it maintains the pH. It tries to maintain the pH, means it acts as a buffer. And when the pH is exactly equal to its pK value, it is having a maximum buffering capacity. So buffer is a solution of a weak acid and its salt, and the buffering range, the range of the buffer is pK plus minus 1, and the maximum buffering is when the pH is equal to its pK value. So these are important points to remember. Now let us quickly revise the points that we have discussed so far. The acid, any chemical species which donates proton. Base, any chemical species that accepts proton. Strong acids and strong bases, almost completely dissociated. Weak acids and weak bases, partially dissociated. Dissociation of weak acids, inversely proportional to the free HN concentration, directly proportional to the pH of the medium, and governed by the Handersen Hasselbalch equation. Buffer, a solution of weak acid and its salt with a strong base or the conjugate base, resists any change in the pH of the medium. Buffering range is pK plus minus 1, and the maximum buffering is when the pH is equal to its pK value. If you wish to better your performance in any of the exams and build a solid foundation for a great career in medical science, please like, share and subscribe our channel Meditut the medical tutorials.